differential scanning colorimetry. So this instrument is mainly used to analyze the thermal denaturation either for the protein or any pharmaceutical products, and also for the engineered protein also we can able to find out by using this differential scanning. So in sample product we are going to find how exactly this protein or else any of your biochemical products will be start denaturing. That study have been easily achieved by using this differential scanning colorimetry. So in 1962, two scientists have been worked under this technique, and they also developed a technique, DSC technique, by Watson as well as Neal, and later on they commercialized this product in 1963 in a conference. So in Pittsburgh conference they have been commercialized this, and initially they have told about it is like an adiabatic process. So we know adiabatic is nothing but there will be a transfer of energy to the surrounding as a work. So that will be you're going to provide that energy in the form of heat. That heat will be go and do some of the work within the sample, and we will measure the difference between it. And mostly we can able to find out the denaturing properties of either protein or biochemical products or biopharmaceutical products, and also we can able to find out the melting temperature. This DSC will completely provide a relative measure of heat flow and also the molecular motion within the component. This will ultimately determine the heat capacity. If you are having a polymer, the heat capacity can be by the polymer, how long it can be withstand. What is the transition occurring on applying this heat? So thermal transition of the polymer can be studied through this, and also for the glass transition temperature, how exactly the glass will be remoduled into some other compound, right? We can find some of the show pieces in our classes, and also in the laboratory classes, in a household for showcasing pieces and all we used to have from the glass, right? So those are being done because of the glass transition temperature. So that, and also the melting point of the each and every component we can able to identify by using this differential scanning colorimetry. So later on they developed this adiabatic differential scanning colorimetry by two scientists in 1964 in Georgia University, and they developed it only for the biochemical activities, biochemistry. In the Department of Biochemistry they have used. Then we will move on to the principles of DSC. So as I told you earlier, it will, on applying a heat, it is going to change the component which is present inside the sample, and it may degrade at a certain point. So if it has reached the melting point temperature, then it will start unfolding or it will start denature because of this applied heat. So you are going to take two samples as well as the reference one. So in sample you will have the exactly which component you need to study it down along with the buffer or a solvent. In reference you are going to use only the buffer or the solvents. So here we will going to maintain the same temperature for both sample as well as the reference in DSC. So there will be an energy change will happen, and also it will maintain a zero temperature. The difference between the sample and as well as the reference will be almost, the temperature is equal within it, and the whole system will also maintain some of the heat which will be transferred within the sample as well as to the surrounding area. We can say if they are using, it usually happen, no, if we have any of the hot pan or any of the hot thing, no, it will start releasing its heat in the form of energy through the outside area. So it will release to the surrounding area. If it is very cold outside, the environment is very cold, the temperature will start decreasing immediately on sending the energies back to the environment. So same thing will happen. If you are having a hot plate, if you are going to keep it down in a metal plate, then the heat will be transferred to the metal plate. So if it is a wooden one, then there will be no any transfer of heat will occurs within the wooden. Only for a certain amount of heat temperature it can withstand. There will not be exchanging of the heat to the wooden as well as when compared to metal, it will be very higher. This technique has been used to identify what is the function of temperature for that given sample, what is the melting point of it, and also we will measure the heat for each sample as well as the reference. This is the ultimate principle of DS. So we have already discussed about the differential thermal analysis in the previous class. So differential, there will be a difference thing between this DSC as well as the DTA. So DSC is nothing but we are going to calculate the amount of heat which is needed to increase the temperature of the sample as well as for the reference. Whereas in the DTA, there will be calculations between the temperature required by the reference and the sample when the heat is kept at the both same temperature. So this will start increasing in the DSC, whereas here they will have a same equivalent temperature itself. So you can find a graph between for DSC. The transition phase will occur based upon the heat released. So you can find there will be an increase in the peak. At the peak point, we can say there will be the melting point can be identified for the given sample. And if it start degrading, then it will have a same curve like structure. You can find there will be a decline phase and then straight horizontal line will be drawn up. So that is represents the transition phases, that is denaturation occurs when on increasing the temperature. So whereas in DTA we will get two curves. So this is what exactly the sample reference in the DSC, and you can find in the right side sample as well as the reference are being taken. So on applying a heat, we are going to observe how exactly the sample will reach on keep on increasing the temperature, how exactly it will reaches the TM value that will be monitored and taken down by the readout devices. So basically in DSC we have two things. One is that heat flux, and which will have a holder as well as a furnace along with that. Then we will have sensor within it, and in thermocouple junction will be attached to the metal so that the voltage as well as the temperature difference can be properly monitored within that. In this diagram you can find this reference pan as well as the sample pan, and there will be a lid which will close so that the entire heat will be completely inside it, and you can find a thermal disc, chromal wire will be attached to the pans, both the reference as well as the sample pan, and we will have a thermocouple junction, and we also have aluminum wire, and also the whole unit will be completely heated, and there will be exist of air, that is outlet will be there for the air which should be coming from the inside, and we also have a thermoelectric disc which will be attached to it so that we can maintain the temperature throughout the whole run of this colorimetry. And we are going to apply a heat, and we will check it out what exactly that is happening inside the sample, because in reference it is like a blank only, and we will use only the reagents or the buffers or the solvents which have been added to the sample. So this is the complete heat flux differential scanning color.